بسم الله الرحمن الرحيم. اللهم صل على محمد عليه الصلاة والسلام. Dear colleagues and Chairman, thank you very much for the invitation. I hope I'll be so clear because giving me a very difficult subject indeed. As you see, very deep sea. I hope I can sail a little bit in this deep sea. We are in the era of omics now. Every cell in our body is controlled by genes. We are in the era of genomics, proteomics, lipidomics. Everything is omics now in our body. In the past, people thought that genes means heredity, but now we are sure that genes mean every function in the body, and every one of us has his own gene identity. The problem is that three things are actually quite integrated: the biology, the genetics, and the biomarkers. I think if you can't see, biology and genetics are so difficult to speak about, but we are surgeons. We are actually interested much in biomarkers because if you cannot see the gene or what's going on, you can see the product of the gene itself, which is the biomarkers. You know that we have now all over the world, the human genome is characterized. There's more than 25,000 genes with very different characters. And sometimes people analyze parts of the gene, which is a nucleotide, which is called nucleotide polymorphism. Now we have two, this precision medicine and personalized medicine, sometimes used as serums each other. Every one of us, of every patient actually, is a disease in itself. He has own genes, own symptoms and signs. In the past, we have our knowledge to treat this patient, but now we have to have some genetic knowledge to treat the origin of this particular patient. That's why this genomic medicine means go into the gene, because as I told you, my dear, that gene control everything. If we come to genetics in the arterial system, actually there is two targets for action: the muscle, smooth muscles, and the matrix, and fibrous tissue. There are separate genes and separate entities for both, but in our work and in practice, we deal with the biomarkers of these, either the muscle or the fibrous tissue or extracellular matrix. Not only the genes that work, but there are some factors that affect the genes. For example, we all know the risk factors, but actually these are outside factors. And sometimes there is much interest in inside factors inside the cell itself. There's maybe some methylation of the gene, deletion, but microRNAs are new jokers. They affect all our genes, our body, and all our diseases. And these are actually the jokers of the genetics nowadays. So epigenetics means other factors outside, either inside the cell or outside the cell, which affect the gene action. I mean the environment around the gene. We have difficult, of course. Important differences are the ethnic difference. European genes are different from African genes. For example, the high cost of the studies is another. The second, because there's a high cost, most of the work is done on a small number of patients because the high cost. The complexity comes from the fact that if we can detect the gene, your next step is to detect which part of the gene is in action, because some part is silencing, some part is regulating, some part is enhancing. We are to go quickly into three entities. And these three entities actually, they are genetically different. The point is the genes of the thoracoabdominal organism are different from the AAA genes, or different from that peripheral arterial disease gene. Maybe some share some genes, but it's very important that the pattern of genetics is different. And second important point, that there is chronic inflammation. Everything, entity of that has some cells which increase the inflammation, some cells which decrease the inflammation. They are all in a case of balanced chronic inflammation. We come TAA. This is the best proof from the genetic point of view, because the inheritance is more or less dominant, and some genes, as we'll see later, are actually recognized like the family. The problem is that after you make, as I say, last line, after I make some exhaustive and expensive research, we find nothing. You find just variants which cannot justify, simply because a very complex subject. Not every time you see a result, but VOS means variants of uncertain significance. What are the biomarkers of the TAA? Some of them, of course, I wrote here some samples of them, but first one is the transforming growth factor. And the last one is important because it is very fixed in all cases of Marfan syndrome. Not only that, it's very well characterized and can be easily detected. Do people need genetic testing or not? Just some indications. Most of part of this is a family history. If you find a family history, we can go into genetic testing. If we find the patient with aneurysm without known risk factors to us, but very important point that it is discovered that there are some protectives of the aneurysm. Some factors inside the biology.
1 minute please.
حاضر. Some خد اكتر خد الوقت كله.
انتوا هي كده القصر العيني واخد وقته مع بعضيه.
So protectives are important because if you see the protectives, you can attack these protectives before the appearance of the aneurysm. Aneurysm TAA may be syndromic or non-syndromic. As I saw, I told you this VSME means vascular smooth muscles are affected in the non-syndromic. The AAA, the family history important because it can be also clear. But some protectives also in the AAA, and some parts are increasing the severity, like elastase one. AAA is very important for many biomarkers, but the most important is the metalloproteins and CRP. Peripheral arterial disease, the genetic factors are weak, not much, but it's discovered that some biological factors are accompanied with severity, homocysteine or endothelin receptors, and the vasculitis are also genetic factors are known. Probable genes in the some candidate here in the peripheral arterial disease. Very important that every step or every action of the plaque is directed by genes, and this every part of the pathology is played by a gene. Now the medicine nowadays, our surgeon nowadays, to go to the gene for prophylaxis and gene for action. The difference of the CBER phone, old phones and new phones. MicroRNA are now the target for gene therapy because they regulate the gene. We can regulate the microRNA to attack the gene. Gene therapy, you are doing it by the stem cells in practice, but now in the era of genetic action, we are aiming at the microvasculature. Everyone can be vascularized, but now not reaching mostly the cells, but that's AI, as Dr. Khaled said, and I think more than. You see, this is my last book about this biology, and thank you very much.
Many thanks, my dear Prof. It's my pleasure to introduce Professor Ezz Eldin Qorashy.